Okay, so let me start again and say that the genetics portion, you have to do to get to 20%. All you got to do is rewrite those questions. The video will be uploaded as soon as I can after today. The protein synthesis, you have to retake the test. So we're going to go over the answers to this test, and you come after school on Tuesday or on Friday and you just take this test. Now, what I normally would do is tell people that if they missed the test, which like you missed the test, right? So I would tell you to do the essay exam. But since I'm giving a retake anyways, you could just take a retake exam, all right? Does that make sense? I have that rule in the syllabus because I just don't want to be unfair to the kids that take the test in class and then you miss it and you come in and take it and then you get some kind of advantage. And I want to offer fairness. So anyways, protein synthesis, DNA and protein synthesis. Could someone close the door, please? The first question is really kind of simple. Either you know it or you don't. If you see that diagram, you see that X-ray diffraction, you got to think Rosalind Franklin. Rosalind Franklin is, without a doubt, is that picture in your mind should be Rosalind Franklin. Don't think Watson and Crick.
They were jerks.
Yeah, they should not have done that. It's unethical, and I was really sad that the Nobel Committee never addressed that. She died. They say again, she died without getting the honors that she deserved. She died of breast cancer and probably because she was exposed to radiation while taking this crystallography picture of DNA. So the fact that this is a picture of DNA, the double helix, and that this helped Watson and Crick develop the knowledge that we have. Now, Watson and Crick did do good work. They did good science. The ethical part of this, though, is an issue. Never well addressed by anybody. So, number 2, which of the following scientists showed that the percentages of adenine molecules in DNA are about to equal to the percentages of thymine? That's remembering that's percentage of A equals percentage of what am I doing? Of T, and the percentage of G equals percentage of C. That was Chargaff.
Chargaff's rule.
And it's Chargaff's rule that in part helped Watson and Crick determine how the structure of DNA and in part how it's replicated. All right. What DNA replication results in 2 DNA molecules? Everybody should know. Number 3 is each new each new. So you have these this DNA molecule, and you're going to replicate it. Remember, replication means you're copying it.
Oh, you were asking.
So when we're making a replication, you're going to make 2 of them. Each of the new molecules is going to have 1 old strand, and then it's going to have 1 new strand. So it's semi-conservative. That's what semi-conservative means. You have 1 old strand, and then you have 1 new strand. Semi-conservative. Remember that repeatedly. There's an experiment, one of the classic experiments in biology, where you were expected to memorize that. Do you remember that? That was that was a long, detailed one that I'm not going to go over, but you had that packet. You did a little class. We discussed it. So those 3 should have been right without a question. If you got them wrong, go back to the drawing board. Make sure you get those in your head. Now, during DNA replication, a DNA strand has this has C, T, A, G, G, T, and it produces a strand with what bases? It can produce a strand that has what? G, A, T, C, C, A. Now, they don't tell you 3 or 5 prime end, but be careful. Just like on a genetics test, you could easily be tricked. All right, let me show you how they can trick you. What if I asked you the same question, okay, and I said C, T, A, G, G, T, and I told you this is 3' end and this is 5' end. What if I told you and I asked you to match it up and I gave you A, I said A, B, C, right? And I gave you 3 prime, let me do it in red. I do 3 prime, and I go G, A, T, C, C, A, 5 prime, and then I give you some other stuff here, and then I do 3 prime, and I do A, C, C, T, A, G, 5 prime. Now I ask you, which one of these would you choose? And I think what happens is 90% of the time, people will choose this one.
Because it's backwards.
And it is backwards, because remember, the problem with this one would be wrong. The problem with that one is that DNA and RNA, when you're transcribing, is anti-parallel. Right? And so you have to match the 3' with the 5', and the 5' with the 3', and that would not match for the first one. But if you take a look at this, if you put this 3' and you attach it to this 5', because that's how it's going to go, then you move backwards, right? So it'd be A would match with T, check. C would match with G, check. C would match with G, check. T would match with A, check. A would match with T, check. And G with C, check. So they do this all the time on these tests. They want to know, do you understand that this is anti-parallel? All right? Are we clear? All right, so don't mess that up, because it's an easy one. But on this particular question, they didn't do that. So which type of RNA are involved in protein synthesis? Well, I hope everybody knows it's 3 of them that we've talked about. There's actually more if you think about it, but 3 is what...
Which RNAs?
The 3 big ones, huh?
You said which RNA?
Which RNA? What 3 types of RNA did I tell you to definitely memorize?
tRNA,
mRNA, rRNA,
and tRNA.
and tRNA. So I told you, definitely memorize those.
Transfer ribosomal.
These 2 are found where?
In the cytosol.
Cytoplasm. And this is in both cyto and nucleus, right? Because it's made in nucleus, but then it goes out to find the ribosomes in the cytoplasm. All right, so let me go ahead now and see if I still have my copy of the universal code here. Yeah. So here's our universal code. We'll talk about exons and introns in a second. We didn't cover it in your last year? No. We did, but I think it was quick. But we went into a little more detail this time. I don't think it's going to be on the end of course exam, but they might put it on there, so I want to always cover it. The chart, I think, was the chart on your end of course exam? Yeah. Was it the round one or the square one? So they'll throw both. So you know, see what I'm saying? So you can see that this stuff is on the test, or at least it was last year. So we'll see what happens this year. All right. So what is produced during transcription? What are you producing during transcription? Let me ask you some additional questions. Where is transcription happening? The nucleus. And you're going from DNA. The message is in DNA, and you have to put it into what?
mRNA.
Right, mRNA. So let's just say RNA. And then you have to take that RNA and change it. You have to make post-transcriptional what we call modifications. What that means is we have to take out the introns. We have to put a methyl cap on it and a poly A tail. So that's what happens. But you don't need to know all that. What's the question that was asked of you? What's made during transcription? And what is that thing? RNA. So RNA molecules. Now, in this question, they predicted some wrong answers. They put RNA polymerase. RNA polymerase is an enzyme that makes RNA, a polymer of RNA. Proteins, it's not the answer. That's what process makes proteins? Translation, right? And of course, what process makes DNA molecules?
DNA replication.
DNA replication. So the only one that fits making RNA is transcription. All right? So let's move on. Oh, now I have to put in a new picture of the universal code.
Are you sure about that?
I might have both of them in here. Let me see if I can find both of them. This would be nice if I could just, because some people have hard times reading one or the other. Yeah, I do. So you could get either one of these or both of these, so be careful. And I've seen people on this mess this one up because they went something like G, they went over here to glutamine for some reason, or they go GA, they do all kinds of interesting things. But let's stick to the right answer, the right way of doing it. So number 7 says, what does AC, let me put a G. What does ACG code for? Let's do the first with the square one, then we'll do it with the round one.
Isn't it 3?
So you go A, then you do C, like somewhere in this square, and then G, which would be across here. So 3, yeah, and in this case, everything is 3, you're right. So this would be 3, according to this one. Now if we do it over here, A, and then C, and then G, we have 3. So always start from the center in the round one and move out to the second row, the second circle, and then move out to the third row. So in both these cases, you get 3, okay? And the answer would be D in that case. Now, I wonder, I can't do this, I can't erase this, but I can delete it, I think. All right, so next is, which one of the following codons codes for a stop? Well, there's only 3 stops. Now, there's 3 stops. 1, 2, 3. You can see the 3 stops here. 1, 2, 3. Right?
But that was 2 yesterday.
I know it was 2 yesterday, but but remember I said I knew it was going to be 3, but I just didn't see it. I agree. That chart, there was something wrong with it. So there's 3 ways of getting stops. There they are. It's either UAA, UAG, UGA. And look here, if you look at the round one, you can have UAA, UAG, or UGA, right? Again, they match, don't they? So you have 3 possible stops. They say, which of the following? Well, they have UGA. Is UGA a stop?
Yeah.
Yes, it is. So UGA was the answer. The rest of those don't give you a stop. They give you different amino acids. It's just reading a chart. Those are just reading charts. The next question is really interesting. I'm going to leave these kind of in the background because I think we might need these again later. So let's just kind of keep them there. The next question had a kind of a drawing that looked like this, and it had a bunch of letters, right? Let me see. You just copy it. Yeah, you have it in front of you. TAC, etc., right? And then over here you had ATG, etc., right? And they labeled this one A. They labeled this one A. And you know what? They did this, didn't they? What is this? What is this stuff here? Versus these are what? These are the nucleotides, right?
Those are like the sugars.
And this is the sugar phosphate backbone. Don't forget that that's the structure of DNA, sugar phosphate backbone. So that's A, they labeled, and then labeled this one B, right? And then they said, okay, well, there's this arrow, right? And then they said, well, here's this thing, this single-stranded, and then you see A, U, G, etc., okay? And then they labeled this thing E, and then they labeled something over here where they put G, C, U, and they labeled this thing F, and then the whole thing they labeled D, and then they had methionine, glycine, phenylalanine, leucine, alanine, and glycine, right? So those were and then they labeled that. Well, they didn't label that at all, but they did say that this was labeled Y. Yes, bless you. I'm sorry, I was in the middle of this. And this is labeled X. I apologize. You're very blessed. I
I'm sorry, I
apologize.
didn't hear you.
So that's kind of the sketch of what they did. And they asked this question. They said, which labeled structure in the figure above, that's this figure, made structure D, made this? First question, what is this?
mRNA.
mRNA. So what makes mRNA? Well, one of these two, what is this thing?
That's DNA.
DNA. Specifically, this is not a question, it's on a test. But specifically, what part of the DNA is represented by this?
Oh, those are the nucleotides.
The DNA that this process is going through, what do we call this part of the DNA?
Replication.
We call this part of the DNA that is replicated, what do we call it? It's a word, simple word, important word. It's the part of the DNA that is transcribed is called
Can I get the beginning letter?
G.
Can I
Gene.
buy a
It's the gene.
Gene.
It's a gene. So this is the DNA that is transcribed is called the gene. This is the mRNA. So which of these two, A or B, is the one that's making the mRNA?
B. Wait, yeah, you wait.
Which one matches with A? Does A match with A?
No, I would say A maybe.
It must be A. A made D. What is the process by which A was copied to make D?
Transcription.
Transcription. So X would be transcription. What is Y?
Translation.
Translation. Taking mRNA to the protein. This is obviously the protein, right? Are we all good with this? All right. So that was that. And let's see if I can delete all this. Oh.
You tried pressing delete.
Yeah, but I'll delete the things I don't want to. Yeah, try that. There you go. All right.
Pretty bad.
Okay. So which labeled structure in the figure above? Okay, we did that. Protein synthesis is determined by the transcription of RNA and translation of. That's right. So translation produces amino acid sequence. So I think we're clear on that. These should have been pretty easy. The sequence of nucleotides in messenger RNA molecules is determined by the sequence of nucleotides in DNA.
Dana.
A change in the nucleotide sequence in the DNA molecule is called a mutation.
A.
And we talked about that yesterday. Please remember you have a quiz on Monday that goes over what that mutation discussion we had yesterday.
Right, me too. I was like, huh? And then you said,
Just know that mutation stuff. Don't be telling me you didn't know it. And, oh my God, what am I going to do?
There's like what point mutation?
Huh?
There's like what point mutation?
As soon as we're done with this, we can go over it. Yes. Huh?
There's like what point mutation?
Yeah.
Yeah, different kinds of stuff.
Stop
Yeah.
now.
wait, so you don't ask us about all the stuff, to, like the frame shift, the nonsense, and
Yes.
the silence?
Yes.
Do we have to know the function? Like,
Yeah, you have to know everything. Anything, everything. There's going to be 10 questions. Boom, you have to know it quick. Multiple choice, you'll do it. You have 15 minutes, 10 minutes to do it, 1 minute apiece. You make your choice. I scan them, goes in the grade book. We're done. A group of friends went to tanning beds for homecoming to get their tan on. Several years later, one developed skin cancer. Was this the result of mutation in what type of cell?
Body cell.
And a body cell. I mean, I think that's clear. When mutations occur in sex cells, they can be passed down to offspring. And remember, that's important. Mutations in DNA can be harmful, beneficial, lethal, all of the above. And by the way, there's kind of a trick here. Let's assume you didn't know that mutations could be beneficial. You should know that it is possible.
Just think about Wolverine.
Well, think about Wolverine, think about the X-Men, yeah. But look here. But here, look at this.
Oh.
It says A is harmful and C is lethal.
Same thing.
Harmful, if it's lethal, it's harmful. So A and C have to be correct, so the only possible answer is all the above. All right, let's take a look at 16. Name two differences between RNA and DNA. I don't know if people misread it, I don't know what, but people gave me all kinds of stuff for that. I think that's super easy. So the first big difference that we talked about is that in RNA you have uracil,
Instead of
and in DNA you have thymine, right? I'm just gonna put T.
in RNA is single-stranded as opposed to
Right, so
DNA.
second, RNA is single-stranded and DNA is double. And I'm gonna go ahead, I only asked for two, but I'm gonna go ahead and give you a third one that you should know. And that's RNA is made with a ribose sugar.
And DNA is made of deoxyribose.
And actually, some people put this, I wanted to give them double credit because ribose has the oxygen.
And D
Not
means
O2.
they took away one of the. Doesn't deoxy still have some oxygen? They just took.
Yeah, they're from a specific place. They took one, there's a missing one oxygen and one carbon. We talked about which one that was, etc., but I'm not gonna. I messed that up. I was like, I almost stuttered there. Deoxy. Sugar. So the sugar is different in both of them. So oxygen missing in one of the carbons. All right. So those are the three major differences. It should have been easy. I don't know why it wasn't for some people, but it should have been. Name two differences between I said that intron exon problem. Now let's go ahead and do this. And this again, this one. For the third time now in two different tests, it hits that whole idea of anti-parallel. You have to understand anti-parallel.
Mendoza.
Yeah,
Can I do this?
yeah. If you'd like, you want to do it on the iPad?
Please.
Do you want?
I was confused with this one.
Can you just put down the DNA?
Well, you'll see in a minute. Let me get this. Hold on. Let me get this right because I get this wrong. It's not going to be Somebody read to me. AGC TC ATG. Go.
You said
Look.
You got to ATG, TGC,
AGC TCA TG.
TG.
CGA, GTCC, TGA, CGC, TGA.
Hold on. TGA. What was it? TGA.
CT.
What?
No, you missed a spot.
CC. I got
Okay, Mendoza, let
CC.
me just check.
TGA, CGCT.
Could someone please?
Yeah, let me see.
No, I just want you to do it right now. Tell me.
Okay.
Let's start again. All right.
So it's AGC,
5 prime AGC
TCA,
TCA
TGT,
T what?
TGT,
TGT
GCG,
GCG
AGT,
AGT
CCT,
CCT
GAC,
GAC
GC,
GC
TGA,
TGA
CTA,
CTA
GG.
GG. It's pretty long. The problem is when I move it now, it's not going to be the same. It's not going to match the part that I say 3 prime. So then we say on this side it's going to be, notice it's 3 prime, and we're going to match it. So you want to read that to me, please? TC.
TCG, AGT, ACA, CGC, TCA,
Hold on. TCA. TCA.
TCA.
GGA. GGA.
CT, GCG, ACT, GAT, CC.
Oh,
ACT, what was it?
GA,
What was it after that?
TCC. And then 5.
All right.
You have to separate those two.
Hey guys, let's try to I know it's taking a little longer than I'd like, but let's try to focus. We got this 3' in there. There's nothing I can do about it, so just kind of work with it. So these two line up, and what you see is, you see a 3' on this end, and you see a 3' on this end. There's a 5' on this end. Then they give you the mRNA sequence.
Just drew an F.
Huh?
Just drew an F.
I didn't. It's messy 3'. All right, so does everybody understand this? Do I not have to go over it?
Wait, let me the problem. No, we finna pay attention. We don't.
I mean, I don't want to waste our time. I don't have to do this.
Please.
So we have this. I had 3' on there already. Got all these random 3' everywhere. It's annoying. All right, so you see these match, right?
Look, the exons are in.
So let me be very clear on this, and let's try to pay attention to what I'm saying, so that we get this straight and we're not repeating ourselves. Gosh, this thing just keeps. So we have this mRNA and we have this DNA, and it just. All right, just go ahead and do this. It's going to look messy, but that's the way it is. Sorry for all the extra 3' because I don't want to rewrite this, so we're not going to do that. So when we look at the mRNA, I'm going to use a different color. We have some methyl group. We have on the 5' end, we have a methyl cap. That tells you that this is the front. Then you have a sequence. You have U, C, A, U, G, U, G, C, G, A, C, G, A, C, G, C, U, G, A, C, right? U, the problem is I run out of space and I have to shrink it, but
There's no way you can make the window wider.
no. GAC, where'd I leave off? GAC, U, A, G, G, A, 3'. So now let me be clear. This part here is the poly A tail.
So the mRNA sequence, this one has to start with.
So listen, look at this. Pay attention. The methyl cap is the head, the poly A is the tail. These two, number 1 and number 2, guys,
She was asking me.
I understand, but guys, look, number 1 and number 2 are done after transcription. So they're not going to be in the DNA. So what are you going to do to try to figure out what part, and the other part you have to understand, the other part, after transcription thing that happens is that this is shorter than this. That's right. The RNA, the mRNA is going to get shorter because the introns from here, somewhere along here, or here, depending on which one is coded, is the coding strand, is gonna that mRNA is gonna be taken out. So your job is to figure out what part of the DNA, what part of this DNA is intron, the part that gets taken out, and which part is exon, the part that actually makes it into the final mRNA. That's your job. Do you mind if I just continue doing this? I appreciate it. So what you need to do, because I promised he could do it, but I just want to finish this and get it over with because it's just so painstaking. So what you got to do is you got to take this, you got to figure out what's the DNA that is in what's the DNA code that's in the mRNA. So what is it? What is the exon? So go ahead and do it. You say U goes with A, C goes with G, you got T, and this is DNA. Remember, we're doing DNA. We're trying to figure out the code that's in DNA. AGT, this is A, C, G, C, T, T, G, C, G, T, C, T, T, G, A, T, C, G, oh, no, C, C, sorry about that. C, C, 8, oh, did I mess that up again? So this A, C, T, A, C, A, C, G, C, T, T, G, C, G, T, G, T, G, A, T, C, C, and that's it. This is the code, right? Everybody see that? That's the DNA code. So what part of this, so which are we going to match it up with? Wilkins, which part are we going to match it up with?
What part of the...
Which of these two RNA, DNA molecules, this one or this one, is the one that's coding for this? It's the bottom one. Why is it the bottom one? Why is it the bottom one? Why is it not the top one? Because it's anti-parallel. Because it's anti-parallel, it's the bottom one that we're going to be worried about. Because you see, the 5' matches the 3'. The 3' matches the 5'. How do we know which one's which? This is a poly A tail, this is the end, and this is the methyl cap at the beginning. So what we go ahead and we do it, we match it up. Where do you see, is this AGT here? No, it's not. Where does AGT begin? Right here, right?
Mm-hmm.
So this is out. So this is used in the, this part's used in the mRNA. Is ACA used in the mRNA? Is it?
Mm-hmm.
Yes, it is. How do I know? There it is, look here, ACA. Then the CGC, is that used? Yes, it is. And then I have a T, but there's no second T. What do I do with that? That's the end of it, right? So that's the end. This part is the exon. This is exon 1. So I have to find the next T, and that's all the way over here. What happens to everything in between? It's out. That's an intron. And then I keep going. I go see T, G. All this is there, isn't it? All the way to the double C, and it's all there. So this is exon 2. And the introns are here. And that's it. Everywhere where the DNA, all the DNA that doesn't make it into the mRNA is the intron. All the DNA that does make it into the RNA is an exon. And that's it. That was the test for the protein synthesis. Oh, there's one more piece that should be pretty easy. And I ended up, I need these two again. So now your question is, what is the sequence of amino acids that's produced by this DNA? And this is asking you to transcribe and then translate. And so this would have been, if you got an essay test, I would just say, here's the DNA, transcribe and translate it. Here's the DNA, here's the mature mRNA. Tell me what the exons and the introns were. You know, that kind of stuff. That would be the essay exam, rather than me giving you the hints that I give you on the multiple choice. So you see there's a T, let's just use white for DNA. There's T, A, C, A, C, C, G, T, G, A, C, T. So there's ACT. This might be an ACT actually. So then to transcribe it, and of course in a problem like this you have to assume that you don't know where the exons and introns are. So you're not going to be required to do take out the introns. So you're going to do the mRNA. So and remember RNA polymerase is doing it. So T goes with an A, then you got a U, then you got a G, then another U, and then GG, and then C, then A, and then C, the U, and the G, and then an A. So of course you have AUG. That's good because we know that AUG is, and we can take a look at it here. A, U, and G gives you methionine, and that's your start. And so methionine is your first amino acid. And that's connected to a what? That's going to be connected to UGG, which is what? Who can do that? U. What's next? G. G. And that's what? That's the tryptophan, TRP. And then that's connected to the next one, which is CAC. It's a codon, as we said. CAC. CAC. That's histidine. And then the next one is UGA, U, G, and A, which is what? Stop. So that's it. Your mRNA, your amino acid sequence, your protein is this, right? You don't have to write stop. You can write stop if you want. If you want to write stop, that's fine, but normally people don't put stop. That's not part of the DNA, that's not part of the protein.
Alright.
Well, you can write it, I wouldn't have a problem with it. I don't think anybody grading an essay on an end of course exam would have a problem with you writing stop. But you do have to, you know, the thing is, that I would separate, I would circle the protein, just so that you know, that people understand, you understand that this is the protein, this is the polypeptide, and the rest is, the stop just means it's not doing any more than that.
Hi.
All right, take it easy.
Hi, Faith. No. I put it.
Oh, high faith for your
I put
sister.
it-
I see your sister, okay, so you can prove that you're here and she's not. I got it. Took a minute, but I got it. All right, so we're good with this protein synthesis? Oh, I'll upload this.